Okay,
Okay.
this is David Zierler, Director of the Caltech Heritage Project. It's Tuesday, February 1st, 2022. I'm delighted to be back with Dr. David Ho. David, as always, it's great to see you.
Good to see you. I have no idea where we left off, but I'll
We're
let you lead the way.
we're going to pick up on when you get to Harvard for medical school, but first, a very timely question. As you know, of course, Moderna just announced that they're launching clinical trials for the HIV vaccine. I'm curious if you're involved in this research at all, if there are any breakthroughs that have allowed for these clinical trials, and if there is any hype associated with the press release, given how difficult creating a vaccine has been for HIV.
Yeah, I think Moderna has a new platform technology, so why not apply it to many things? And HIV is one of those intractable problems, right, for vaccine development. So it makes sense to try. Do I think it will to overcome the challenges that we have faced for several decades? No. Uh, it You're showing the immune system the same molecule, just using a different delivery strategy. But the fundamental problem is with the structure of the HIV envelope protein. So it's something that must be tried to see if you get an unexpected result, but the expectation would be that they will face the same structural problems that the field's been facing all this time.
Now, when you say a new platform that Moderna has, what does that mean?
Well, that means, you want to show the way we showed the spike of COVID-19 virus to the immune system is now largely using viral vectors or mRNA, right? So those are just the delivery platforms. Or you could give the protein itself. That's another way, right? But if you give the RNA will make the protein, and the protein will be presented to the immune system. If you use a so-called viral vector like a Johnson & Johnson vaccine, that delivers the gene to the muscle, and the muscle will make the protein and show it to the immune system. So it's all but the fundamental problem is that molecule that's then presented to the immune system is covered with sugar, covered with variable sequences, so that the type of immune response you elicit will be very, very narrow. And HIV is, all the viruses out there are rather diverse. You could protect against the version that you show to the immune system, but you won't be able to protect very well against other versions. And that's the problem. I don't think that's a platform problem, but they're using the platform to see if that will help. It's the logical thing to do, but it alone will not overcome the problem.
It's a moonshot in the sense where it's very much worth at least trying, is what you're saying.
Yeah. It has to be tried.
So let's say there's a miracle and it works. What would that look like? How would it work? What would be different now than all of the work that's been done trying to achieve a vaccine so far?
Well, that would be close to a miracle if it worked, and then we really have a solution, right, to the pandemic. You would try to scale that up and administer it initially to all the high-risk people. You have to do the trial to show that it indeed protects. That's going to take many years, by the way, because this is not COVID, where cases are accumulating so rapidly, so you could have the end result read out very quickly. This will be years of trial, in development, years of trial. So even if the strategy works, I think the vaccine is 5+ years or more away.
Well, David, let's go now back to Harvard. The first question there is, were you open to the idea of pursuing a medical degree to becoming a medical doctor? Or did you know even from the beginning that this was the step that you wanted to take on your way to a career in fundamental research?
I think there are times where in you vacillate. But largely, I was leaning toward a research career in an academic center, and probably, one focused on important medical diseases. And over the years, my interest within that area had varied from something as diverse as cardiology versus infectious diseases.
David, I'm curious, coming to Los Angeles, being at Caltech, how widely traveled were you in the United States? When you got to Cambridge, had you seen other parts of the country before?
I've seen parts of California driving around. Have I gone? No, I've not gone to any other place. So outside of California, we talked about this. I've never traveled outside of Taiwan, and then coming to the U.S., going through Japan and Hawaii. That was the first flight I ever taken. The next flight was some years later to Cambridge.
And what were your impressions of Boston when you first arrived? What did you think?
It's very different from LA. It's much smaller. I had a bike and I could get around Boston, Cambridge on the bike. And it was very efficient. People drove like crazy people in Boston. LA, I think, by comparison, was very law-abiding when it comes to driving. Well, Boston, there are lots of crazy drivers. And Boston is dramatically different because it's such a college town with so many schools. So the whole population seemed young. And then the reality of a winter in Boston hit sometime later.
David, did you have any opportunity for lab work in those first few years, given how important the research was to you, or was it all classes?
No, there was no lab work for the first two years or so. They were all classes. And there's really only any serious research work only occurred in the fourth year of medical school. The schedule is so tight. There's hardly time for anything. And so, and then when I got to the fourth year and tried to do a thesis, it's only a course of a few months. So you can't get anything serious done in the laboratory. So it turned into sort of a, review of a field, summarize the current status and that sort of things. Nowadays, it sounds rather superficial.
I'm curious if in graduate school, all of the revolutions that were happening in biology, reverse transcriptase, recombinant DNA, if these things were registering with you in medical school as opposed to being in a PhD program for biology.
Yeah, they were registering. I think, I was at Harvard where, research is emphasized. And I was in a program called Health Science Technology that's joint Harvard Medical School and MIT. And so we had our courses be more quantitative, more research oriented, and more cutting edge topics coming up. And so I got a lot of exposure to, when you do the biochemistry in medical school, that had a lot of modern molecular biology in there.
What were some of the big questions that you wanted to pursue besides just doing well in the classes? What were you hoping to get out of medical school in gaining this education?
Yeah, it was not so much focused on a particular discipline, but when you go through the medical textbooks, you realize that we knew a lot about anatomy, physiology, But then when you go into diseases, many of the diseases remain totally unexplained, right? How, you look at, yes, some of the infectious diseases are obvious. We know what the cause is. And fortunately, for many of the bacterial and fungal infections, we already have treatment. But for viruses, there was hardly any treatment then. And then for many, say, neurologic or rheumatologic diseases, we sort of understand what mechanism may be at play, but the fundamental cause was unknown. And for many of them, still unknown today, right? Yes, we know Alzheimer's are associated with these plaques, but how are they, what triggered them? And various other neurodegenerative diseases, they're still mysterious. And something as common as multiple sclerosis, we still don't know what the cause is. Yes, and maybe triggered by infection, followed by some autoimmunity. We have some vague notions as to what might be going on. Same thing with lupus or arthritis, rheumatoid arthritis. The body is attacking itself, but what triggered it? We still don't know. So what intrigued me as a medical student was, well, what about these remaining medical mysteries? How do we understand them? And finding a cause, right? And because the ultimate solution is understanding what the fundamental cause is. So that's what I was interested. But, in terms of, for a moment, I was interested in multiple sclerosis. Next moment, I was interested in something else, depending on the course I was taking at the time.
And what were some of your more enjoyable courses and formative influences from the faculty, the professors at Harvard Medical School?
Yeah, I think I enjoy courses that were more quantitative, and that's because of coming from Caltech. That's more familiar to me. So for some of the cardiovascular courses or pulmonary, it's very mechanical. And, there's either fluid dynamics or air dynamics. And the track I was on at Harvard Medical School was very quantitative. So it brought a lot of those type of applied physics and also mathematics to those disciplines. But in the end, I actually was more interested in solving mysteries, it's almost like solving problems that we spoke about for Caltech classes or Caltech exams. I wanted to solve the medical mysteries that persisted till then and persisted till today. And this is why I know you haven't gotten there yet, but that's why I jumped on HIV, because that was an obvious medical mystery that we were seeing for the very first time.
What were some of the big questions in virology and immunology from your perspective in medical school? What was the frontier of knowledge at that point?
Yeah, I think one aspect that really started to take root in me as I was doing my medical school training as well as subsequent training was the episodic outbreaks of infectious diseases. And initially they were all mysterious, right? And so what colored my whole experience was, for example, Legionnaire's disease. So these legionnaires congregated in Philadelphia at a convention, and, X number of them came down with pneumonia, all more or less at the same time. It was an outbreak. The CDC, jumped into action, trying to solve that mystery. It's like a detective story. And yes, over time, they did solve it, and it was a new bacterium, now known as Legionella pneumophila, right? And that for example, stuck with me. And it was a really good case study of a medical mystery that actually puzzled the world, and then with good detective work, it was figured out, and a new organism was discovered. And you could say, okay, Lyme disease, okay, that also popped up around the same time period, right? People have rashes and have arthritis, and coming up episodically and due to a tick bite, in many cases follow a tick bite. What's going on? And that was not known until the new spirochetal bacterium was discovered. And in many ways, one medical mystery at that same time, which wasn't solved till later, was what we used to call non-A, non-B hepatitis. So these people, it was clear that it is a transmissible disease. Drug addicts were getting it, blood transfusion recipients were getting it. So it's in the blood, but we can never identify. But it's not A, it's not B. And today we call it C, but that was discovered, and we still cannot grow that virus in the lab, but using molecular techniques, that was figured out later. But that was a medical mystery to me, and that would actually formed a very strong foundation for me to want to pursue these, mysteries that turn out to be infectious diseases. And I also have to tell you, just some years prior to that, even hepatitis B was just being discovered. And that is also a virus we cannot grow. But, a Nobel Prize was awarded for the discovery of the Australian antigen. That is hepatitis B proteins. And so we had a marker, we had a handle on the virus, even though we cannot grow the virus. And then, of course, seeing how then that information led to the development of hepatitis B vaccine, and that allow people to prevent that infection, and then consequently, years later, shown to prevent liver cancer, for example. So infections coupled with cancer, exemplified by hepatitis B, exemplified by herpes simplex with cervical cancer for women. Those linkages also formed a very important impression in my mind.
David, what about the role of genetics versus environmental factors? Diseases that you get that were passed down to you versus diseases that you get because of things that you're exposed to. What kind of education or training did you get in that regard during medical school?
Not a lot, but the classic one is sickle trait, right? Why would sickle trait be selected when it could lead to inheriting two sickle the genes that result in such devastation for the child. But it turns out if you're so-called heterozygous, so you only have one bad copy of the gene, you're actually somewhat protected for certain forms of malaria, which is so common in sub-Saharan Africa. So it's a clear example of genetics affecting a very important infectious disease. That one stood out. And of course, now we know there are many genes, particularly genes that involve the immune system, that affect clinical outcome differently.
After you had completed all of the required courses, what opportunities were there at Harvard for specialization within medical school, either in a laboratory setting or in a clinical setting?
Yeah, in medical school there wasn't much. Although I failed to mention that prior to medical school, I joined an MIT lab uh, for several months. This has been between graduation and start of medical school, and I worked in a antibody lab, and that formed a very strong foundation for me because I still work on antibodies to a large extent. And that was a lab led by Hermaneisen, who is a distinguished immunologist, a leading figure in antibody function and structure.
Did you keep up that relationship during medical
Yes.
school?
A little bit, but amazingly, decades later, uh, Hermaneisen and I would communicate by email and by phone on topics of mutual interest. He's several decades my senior, but we I was able to make a full circle with him and reconnect, mostly on HIV antibody issues.
What were some of the big questions in his lab and in antibody research in general at that point?
Yeah, a lot of antibody structure and understanding which portions of the antibodies are important for recognizing the target. So structure and function of antibodies and how antibodies were made in the body through gene recombinations. That aspect, which is very exciting, was not known back then. And it took, another MIT professor, Susumu Tonegawa, to figure that out some years later. And of course, he won the Nobel Prize for that.
At what point in medical school do you start thinking about next stages, either residency or career moves? How does that work?
Yeah, I think you start thinking about that once you finish your basic courses and you're going through your clinical rotations. So the major rotations are, for example, internal medicine, surgery, obstetrics, gynecology, pediatrics, and so on, right? And typical of any medical student, you get interested in whatever you're rotating. So your interests evolve through that third year. By fourth year, you're forced to begin to narrow down which area you want to go to. And for those who have a research career in mind, you tend to go into internal medicine first because that's more of an intellectual discipline where you trying to understand a problem and get at the root of the medical problem. Or you could go into pathology. That's another area where academic medicine excels. You wouldn't want to be spending 5-6 hours a day in a surgical suite, right? So that's although there are a number of people who pursue research from that track, but I would say large majority come from internal medicine and pathology. So I gravitated toward internal medicine and still not sure which area I would pursue. And that's when, I was tossing around the idea of infectious diseases because I had by then, a sneaky suspicion some of the mysterious illnesses were due to infections that we still have not uncovered. One classic example is peptic ulcers. We used to think it's too much acid, right? Years back, but it's due to a bacterial infection, right? Campylobacter. And somebody from Australia won the Nobel Prize for that discovery. So I was tossing around, cardiology versus infectious diseases by the time I was getting into medical training.
David, just anecdotally, among your classmates, how unique were you in terms of having aspirations in a research career and not a clinical career?
It's not unique. I would say about 25% of the class has such aspirations. At an institution like Harvard, that may be skewing high because it's Harvard. If you had gone to a state medical school in the middle of the country, that proportion would be much lower, I suspect.
Did you have any opportunity to interface with patients at all during medical school?
Oh, sure. During third and fourth year, when you do the rotations, you're doing that in the hospitals, interacting with patients, although supervised by interns, residents, and attending physicians. But the patient interactions begin in the third year. Nowadays, they begin much earlier, but I'm talking about my experience.
David, communicating with patients, literally putting a human face to the conditions that you were thinking about. In what ways was that useful in your intellectual development?
Well, you get to appreciate clinical problems. You hear it from the patients. You have that raw understanding of what conditions really mean to a particular individual, right? And then you have to interact with patients, so you learn those communication skills, and you learn to be empathetic. You learn to convey good news as well as bad news, and then when you take it back to the discussion, you're then all of a sudden more focused on the problem and how to solve it, or how to diagnose it. And so it's really a very interesting experience that you live through for several years, and actually it really helps to get a deep understanding of the disease. And that's why for some conditions, it's really hard for straight PhDs to do medical research unless they have a pretty good grasp of that. I mean, they could learn that along the way without an MD degree, but having a full clinical background is really very, very helpful in addressing clinical research questions.
When you graduated from medical school, what were the opportunities available to you at that point? What did you want to do next?
Well, I chose to train in internal medicine as a sort of stepping stone to an academic medicine career. And as I said, it was internal medicine with the idea of ultimately going into infections or cardiovascular diseases. And at that time, I was interested in the infection. That part is clear, but the cardiovascular diseases, there was a lot of excitement going on in terms of understanding lipid metabolism and cholesterol and how they may lead to atherosclerosis and, huge clinical problems that require new solutions.
And what were the residency programs at that point that were considered at the cutting edge of internal medicine and were doing the kinds of things that you wanted to get exposure to?
Yeah, I mean, there are probably a couple dozen programs throughout the U.S. that are considered top echelon, and you could get good training at any of them. And they're all at academic medical centers, right, in the big cities. Boston, New York, Baltimore, D.C., St. Louis and Chicago and San Francisco, L.A., Seattle, and you hope to land in one of those big programs and get good clinical training and also get some exposure to cutting edge research that's addressing unsolved medical problems.
And for you, UCLA was also an opportunity to get back home to some degree.
Yes, and to get away from the winters.
Tell me about getting to UCLA. Obviously, it's a much bigger place than Caltech. What was it like there when you first arrived?
Well, you arrive as medical interns who, while you have gone to medical school and you now have an MD degree and you're allowed to wear the white coats and walk around and call yourself a doctor, you still are in training and you're the lowest physicians in the totem pole. And so, I think it's probably the scariest period for a doctor, because in medical school, you have supervision. You could always lean on somebody. But once you're on your own, even though you still have people who supervise, they're giving you a fair amount of latitude. And so that responsibility felt very heavy, at least in the beginning, until you settle in. And that settling period could be a few weeks to a few months, and for some, a few years. And so it's a pretty frightening period for a physician.
And what was your first project or rotation that you were on?
Believe it or not, I got thrown into the ICU as my first rotation.
Wow.
And it was the cardiac intensive care unit. And, it's every 3 nights. Every third night, you're not getting much sleep. So that's exhausting. And then you're taking care of the sickest patients with cardiac problems. So you see many die. But you appreciate what good medical care could do also. You see people who basically have died, and you resurrect them and you get them back out of the hospital and back to life. That was like being thrown into the deep end of the pool and forced to learn how to swim on the spot. Yeah.
And I'm curious, in that environment, what are you learning from the nurses and what are you learning from the attending?
You learn from everybody. Even though you could write the orders for the nurses and they have to execute your orders, most of the nurses have been there for 10 plus years. So they know what to do, and you learn from everybody, including the nurses. And but they, obviously, on rounds discussing with the attending physicians and senior residents, right? You learn from those people as well. You pick up very quickly, and within weeks you could speak the language. You could you learn how a hospital functions, how to order things, how to run to the right places to look for X-rays and what have you. You learn some of the basic procedures very quickly, so you could physically carry out certain things like putting in lines for patients and putting in certain catheters. And so it's a very, very steep learning curve for the first 6 months.
What was your rotation after that, after ICU cardiology?
And then the medical wards for several months. And there you see many different diseases, right? From acute ones to referral cases to terminal cancer. And naturally, I gravitated toward those diseases that had unknown causes.
Such as what?
Like, for example, a patient comes in with 3 months of fever, what we call fever or unknown origin. What's going on? Is that an infection that we haven't uncovered? Is that a rheumatologic condition that we haven't diagnosed? Or is it something else, some genetic disease, depending on the age of the patient, obviously. But those are the fascinating cases.
Were there mentors during your residency that you really learned from, their approach to the research and to working with patients?
There always are mentors in the ICU, cardiac ICU, or the medical ICU, or on the medical wards. Especially in the first couple of years, you just pick up, nuggets, or what we call clinical pearls from various attending physicians as well as peers. So it just comes in random forms. It's not as systematic as class learning. Yeah.
What opportunities, if any, did you have for laboratory research during your residency?
None. I mean, you, especially in those days, you just schedule from one rotation to another, and there's hardly any time to sleep, not to mention research.
So basically, internal medicine gives you the widest possible angle to every malady that humans face when they get to a hospital.
Right, right.
What was valuable to you to have such a wide perspective on all of these issues?
Yeah, it is really to understand a wide range of diseases and some of the underlying pathologies or the mechanism of, what we call the pathogenesis, how certain diseases come about. Obviously, some are known and some are unknown. And getting a really good appreciation of what could go wrong in a human body, right? Or what organisms are out there that could invade us and cause problems, or how our immune system could attack our own body due to some signal that triggered it, which we to this day still don't understand fully. So those are just a few of many examples of what you learn to appreciate through clinical training. And then, you decide which sort of problems interest you the most. And for people who eventually turn to infectious diseases, it's because the long history that many of them turn out to be treatable, right? It wasn't so until the antibiotic era, of course, after Fleming and so on. But at least by the time I was at that stage in my career, many infections were treatable. Many bacterial infections were treatable. A number of fungal infections, a number of parasitic infections were treatable with drugs. So it's a bit unlike cancer, most of which were untreatable at the time. And many are still untreatable today.
David, the last topic we'll talk about for today's discussion, we touched on it briefly in our initial conversation, but now that we're in the narrative, we can go into it a little in more detail. Do you have a specific memory of when people started to come into the hospital in Los Angeles with a new illness that would later be identified as an HIV infection leading to AIDS? Do you have a specific memory of that?
Yes, very clearly, because this was in my senior residency year, so it would be late 1980. Okay, so remember, 81 was the year that AIDS became officially recognized. So this would be a period before that. So it's late 1980, a gay man comes into the hospital with, bilateral pneumonia.
What does that mean, bilateral?
Both lungs infiltrated with something. causing pneumonia, having a hard time breathing. And on X-ray, you see haziness on both lungs. That was already unusual in a young person. And then obviously no obvious diagnosis was made until a bronchoscopy was performed. So you put the tube down, you wash the lung, and you take the fluid out. And from those washings, it was determined that these people had that patient had Pneumocystis carinii infection. That's a infection you only see in people whose immune system have been destroyed.
And that was known already, that this was unique in terms of what it was doing?
So prior to that, Pneumocystis carinii pneumonia was seen in patients on chemotherapy and transplant. And this first case had no such medical history. So that was extremely unusual. And then he also had seizures. And when you do a CT scan of the brain, there were holes in the brain. And of course, we had to find out what was the cause, and then you you do a brain biopsy, a small opening brain biopsy, and it turns out to be a parasite named Toxoplasma. And that too is an infection that you only see in cancer patient or transplant patients. So that already, even in one case, that raised the suspicion that this patient had some underlying immune deficiency. But this person had lived a life of 20, I forget the age, 20, 30 years without any problem. So it's not a genetic immune deficiency.
David, when did it start to be apparent? Where were the patterns that you recognized that this illness, before it was even identified as AIDS, was harming gay men in particular?
So that's you asked the first case. So that's the first case. And then he was treated, he went home, and he died within weeks after discharge.
And you knew he was gay? How did that become known? You asked him?
Oh yes. I mean, it's common that we take a sexual history as part of a complete medical history. And what was striking, and this will be very important, is that he had a number of what we call gay-related infections. So he had syphilis, he had what we call gay bowel disease. So it's essentially, gonorrhea of the rectum or chlamydia of the rectum. And so even from the first case, we knew that there was something wrong with the immune system and that this person was a gay man who had a number of infections. So that automatically, it doesn't take much to think, well, could what's going on here be a new infection? But that's one case. And then a couple of weeks later, another gay man came into the hospital. Now, not with exactly the same syndrome, but certainly with pneumocystis pneumonia. And then had retinal infection by a virus called CMV. And that you also see in transplant patients. So now you have two cases. And then a few weeks later, there was another one. So in this way, 5 cases were seen in late 1980 and early 1981. And those 5 cases, I happened to be there. Okay, I was not in charge. I was a resident, so I wasn't an intern, but I was a senior resident. And by then, I had enough medical knowledge to know that there's something wrong here. And all of them were gay men with a number of gay-related infections, and they all had a similar syndrome that suggests their immune system was impaired. And they were coming in one after another, not rapidly, but over a few months' time. And then there was a common thread. They were all gay. They were all young men, and they had no prior history except for the various sexually transmitted diseases. And if you take a careful medical history, they had lots of partners, sometimes into the hundreds. So infectious transmissible disease had to be on the list. But then if you look at the textbook, is there a transmissible disease that knocked out the immune system? No. So even to a nascent physician, it didn't take much to say, could this be something new? And that's the mystery part again, right? I mean, to me, I was just tracking these cases, even if they're not mine, right? I would find out whether there's more such cases coming into the hospital on the nights that I'm not on. And then fortuitously, by June of 1981, when I became chief resident, then I hear about every single case admitted to the medical service, because the interns and residents had to report to me all the cases. And from 1980, well, by then, the first 5 cases have been summarized and reported to the CDC. And CDC have been noting that there's been a dramatic increase in requests for pentamidine. Pentamidine is used to treat pneumocystis pneumonia. And so CDC made the connection of these 5 cases with the request nationwide for pentamidine. That's a control drug at the time. So CDC would hear about every case for pneumocystis that way.
David, do you have a sense that going back to that very first patient, was this patient 0 or close to it? In other words, was this happening earlier than anywhere else except perhaps maybe New York?
Yes, I'm sure it was happening in New York and San Francisco. And of course, after the first report from LA, New York and San Francisco, within a few weeks, had similar reports, all through what's called the MMWR, the Morbidity Mortality Weekly reports from the CDC. And the finding from UCLA, this is not my finding, from the immunology lab that also provided a common thread for all of them, is by 1981, there was a new test that measured certain type of T cell counts, particularly CD4 T cells. And that number was found to be low for all of them. So that really unified this cluster of patients, all gay men, all seemingly immunodeficient, and documented low CD4 lymphocyte count. And then having a multitude of infections that consistent with having severe immunodeficiency. And then all of them had a normal history, medical history prior to that. So it was not a inborn immunodeficiency disorder. It was acquired, right?
David, for my last question, I'll just note that for you, who was interested in the mysteries of infectious diseases, boy, did you find one. And my question is, with all of your appreciation for the value of being in a clinical environment and the human dimension here, which is, it's scary enough to have a life-threatening illness, but it has to be even scarier when the medical professionals have no idea what it is. Did that plant a seed in you that, looking back, you recognize that was the additional motivation that would compel you to spend your career, your life working on this?
Yeah. Well, as you could tell, I was drawn to find out what was going on with these few patients, and I have to emphasize few, right? So, and at that point in time, I was leaning toward cardiology, even though I had these interests in infections. And people, and cardiology is a very dynamic discipline. And I have many mentors who say, why would you want to give that up to focus on this esoteric problem? But, you know, I have to say the mystery part really captured me. And you know, I just pursued it. I just knew it was this important scientific problem. You have a new transmissible disease, a seemingly transmissible disease that destroys the immune system. Whether it's esoteric or not, it's an important scientific question of what's doing that. And that's why I went into it. And I was all in even before I joined the lab from the clinical side. And I spent a year collecting such cases from middle of 1981 to middle of 1982. And then I returned to Harvard and went into infectious diseases and went into the lab to study this problem.
Well, David, on that note, we'll pick up for next time where you get further and further into this mystery.